Part 3 is stem cells. So stem cells are undifferentiated cells that can then differentiate it into different types of specialized cells. So there are two types of stem cells you need to know about in humans or animals, and that is embryonic stem cells. So the embryo is when the sperm has fertilized the egg, and the egg then starts to divide. So they are undifferentiated cells. They have the potential to turn into any kind of cell. The adult stem cells, they are found in places like your bone marrow, so they can only turn into some types of cells such as blood cells. Now, you do have other adult stem cells around your body, but they're very, very limited to what type of cells they can produce. So here's an embryonic stem cell, and you can see it has an inner cell mass, and then the cells will eventually, those undifferentiated cells, they will differentiate into any type of specialized cell. So in this case, fat cells, nerve cells, red blood cells, white blood cells, lipids, anything. So they can produce any type of specialized cell. Whereas an adult stem cell, so for example, blood stem cells are found within the bone marrow of your bones, and they can differentiate into different types of blood cells. So they're specifically making different types of blood cell, in this case red, white blood cells and platelets. Now, why are stem cells so useful for us in the future and at the moment? So you could do something called therapeutic cloning. So this diagram is showing therapeutic cloning with embryos. Now, embryos can be harvested from unwanted embryos. So for example, after fertility treatments, some people might have spare embryos that they don't want anymore, and they're going to be thrown away. So they can then culture these to produce more stem cells. Chemicals are used to make them turn into different types of cell, and this is obviously at the research stage, where you can produce muscle cells, nerve cells, possibly insulin producing cells to cure people with diabetes. So they could treat many different conditions and diseases. But the key ethical question we need to think about is it right to destroy an embryo to get to the stem cells? So scientists have thought about this, and they've discovered a new way of using adult cells and adult stem cells and making them behave in a way like an embryonic stem cell. So what you can see here, you can see a human egg cell. So it's not an embryo, it's a human egg cell. And what they do is they remove the nucleus, and then they take the nucleus from a patient cell and they transfer it into the egg cell, and they stimulate the cell to divide, and then the cell develops itself into an embryo. And then after 4 or 5 days, you can remove those stem cells. They culture them, that means they grow lots of them, and then they could be injected back into the adult patient to try and cure a specific condition. Obviously this is all research at this stage, but there's a real advantage here, and the big advantage is that you're using an adult stem cell. You're not destroying an embryo, so people can give their consent and say that they are happy for their adult stem cells to be used. In exams, they like to give you lots of information about stem cells, and there are different arguments for and against doing stem cell research, particularly with embryos. So these are a couple of arguments for and against. Obviously, I think there are more arguments, and we'll look at that when we go through an exam question. So for stem cell research, it's curing patients. You're curing patients with stem cells. Is that more important than the rights of embryos? So actually trying to relieve suffering of people who are living. So they're just using unwanted embryos as well, that might be from fertility clinics that would normally be destroyed. Those against stem cell research would argue that embryos are human life, so you're destroying a life. Scientists could also find other sources of stem cells, for example, using adult cells in therapeutic cell cloning. So stem cells are also found in plants. So they are found in what we call the meristem. Now, there's a lot of information here. You don't need to know all of this information. All you need to know is that the meristem is basically the areas of growth in the plant. So wherever there is growth, so the top of the stem, for example, to produce new leaves, or in the stem itself, or actual growth within the roots to make the roots longer. So they're able to produce clones of the plant. They can be used to grow crops with specific features for a farmer. So, for example, disease resistance. So you could then develop a plant with disease resistance, then clone that plant through taking cuttings or doing something called tissue culture. Okay, so we're on to the question checkpoint. Again, as normal, what I would like you to do is to have a good read of these, and I'd like you to pause and have a go at answering them. So 3, 2, 1, pause. Okay, so let's look at the questions. So first one is quite simple. So stem cells are undifferentiated. Which statement about stem cells is correct? And you literally need to go through the list, and you'll find that actually meristem cells in plants can differentiate throughout the life of the plant. It's a case of going through each one and seeing which ones are completely incorrect, and therefore you can find the correct one. Okay, so this one here is more kind of what you'd expect to see in a higher tier paper. So they give you lots of information about stem cells, therapeutic cell cloning, and they're talking about advantages and disadvantages. So I'm going to show you the answers for advantages. Now, you only need 2 of these answers, okay, but you could write these down maybe with a green pen. So advantages are that you could cure or treat disease. So you don't need to have in your answer both cure and treat disease. Just one of those would be fine. It also replaces cells, tissues and organs. Cells are unlikely to be rejected because they're your own cells, so your white blood cells are not going to attack the new cells because they are your cells. And obviously, if you're waiting for a transplant, if they could grow spare organs, then you would not have to wait for a transplant as long. Disadvantages, obviously, with embryos, if you use embryos, life is killed or destroyed. There might be a shortage of eggs or donors. The key thing is we don't know what the risks and side effects might be. Okay, and also a key thing here is if the cell contained a viral infection, you actually can transfer that viral infection into those stem cells. So you could actually transfer viral infections, which is quite severe. It could cause you lots of problems.